Okay, we're back again. So we have learned a little bit more about what antibodies actually are, their shapes, how unique they are, each individual different type of antibody that your immune system can produce, how very, very specific they are for particular types of antigens. Well, now we're going to talk about, well, what causes a B cell to get activated and to specialize into those plasma cells that actually produce antibodies. Okay, so as we talked about before, here's a typical B lymphocyte, and the surface of that B lymphocyte or B cell is going to be completely coated with B cell receptors. And remember, we also talked about how those B cell receptors are actually IgD type antibody molecules, but they're just embedded all over the surface of that B cell instead of floating around in body fluids. And again, you know, these diagrams are very highly embellished. The size of a B cell receptor is not going to be anything like that at all. You would not be able to see those B cell receptors sticking out of the membrane of a B cell. They're proteins, which are much, 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 much smaller than a cell. Now, over here, this diagram is blowing up a little chunk of the membrane of a B cell and showing you just one individual B cell receptor embedded there within the surface. And you can see when you look at that, indeed, yeah, it looks just like it's a Y-shaped antibody molecule that is embedded there in the surface. So your immune system is capable of generating, each person, you may have at least 1 billion different types of B cells produced by your immune system. Now here's something that's really, really important. Different B cell types have different B cell receptors embedded in their surface. And all of those receptors on one particular type of B cell will bind to only one antigenic determinant, one epitope of one antigen. So this little B cell here, all of its B cell receptors might bind an epitope of an HIV antigen. Then you got another B cell over here, and all of its B cell receptors are going to bind to one epitope on a Salmonella enterica bacterium surface, etc. Okay? So one B cell is not going to have receptors that bind to multiple different types of antigens. Okay? And that's what they're showing you over here as well. The antigenic determinant of an antigen binds down into the pocket, the antigen binding site, the variable region of the B cell receptor, just like we talked about with an antibody molecule. Now, since we have so many different types of B cells, what that means is no matter what antigens you get exposed to, you're probably going to have one or more B cells that located in your lymph nodes, your tonsils, those other lymphatic tissues that if they encounter their corresponding antigen, you're going to have one somewhere that can respond to it, one or more, hopefully more than just one. Okay, but again, this is key. Each individual type of B cell, recognizes only one antigenic determinant or epitope. Okay, so we're going to talk more about how B cell activation occurs, because when a B cell, like this one over here, by the way, this was supposed to represent an antigen of some type with epitopes in it repeated over and over again. And so notice that you can have multiple B cell receptors binding to those same epitopes on an antigen. But So B cell binds to its corresponding antigen. That does not mean that it's going to immediately start secreting and releasing antibodies into your bloodstream, your lymph, and your tissue fluids. There's actually a multi-step process that has to take place. And this is kind of a cartoon view, as you can tell, of those different steps. So here's the first one. This is our friendly little B cell right here, and these are the evil bacteria with antigens in their surfaces. And now what they're showing you here, so you've got one B cell. It's going to have one type of B cell receptor in its surface, and you got the same type of bacteria here, but any bacterium is going to have multiple types of antigens in its surface. Well, only one epitope on one of those types of antigens will bind to the receptor in the B cell. Okay, and if this B cell gets lucky, finds its corresponding antigen, that's the very first step in it becoming activated and starting to produce antibodies. Some other things have to happen. One thing is, this is not enough in most cases. There are some cases where it is. But in most cases, another cell has to come along, a type of T cell called a helper T cell, that has also been activated by that same antigen. We'll talk about how that happens. And then it has to send signals to this B cell telling it, yeah, go ahead and convert into plasma cells and start producing antibodies. So what that means is you've really got to have two different types of cells that have been activated by a particular antigen in order for antibodies to actually be produced. And that's a good thing. It's a safeguard. It helps prevent, let's say one of your B cells made a mistake and bound to one of the surface antigens on your cells. If it didn't require that second type of cell to also bind to that same antigen and get activated, then it might start cranking out antibodies, which would then bind to your own cells, and then all those nasty things that we talked about antibodies doing could happen to your own cells, and that would not be a good thing. So that's kind of a safeguard type thing. A B cell must receive help from a helper T cell in order to actually crank out its antibodies. And then, as we'll see, the B cell over here that got activated by its antigen and received help from this T cell will start multiplying over and over and over again. And as those cells keep multiplying, many of them are going to specialize or convert into plasma cells, and then others are going to convert into memory cells. The memory cells are the ones that are going to hang around for a long time, even many, many, many years. And if you ever get exposed to that same organism, that same pathogen again, they'll very rapidly be able to start generating antibodies to try to intercept it. Okay. Now your plasma cells over here, they're going to start secreting antibodies, releasing those into the bloodstream, into the lymph. They'll get into your tissue fluids, especially if they're IgG. Then those antibodies will float around and find other examples of that same type of microbe with those same antigens on their surface. And then that's going to help your immune system get rid of these guys by using those different methods that we talked about in the last video clip. And again, one of those methods is to make it more likely that phagocytes, macrophages, are going to engulf them and digest them. And then finally, here's your memory cell over here. So as we'll see a little bit later, in the future, if these same little critters get inside your body again, that same antigenic determinant on its surface, that same epitope, will bind to the B cell receptors on the memory cell, and that memory cell will very rapidly get activated and start producing antibodies to help you get rid of it. Okay, another very important thing. The receptors in the surface of the B cell bind to a particular epitope on an antigen. When that B cell gets stimulated, starts making plasma cells, the antibodies produced by that plasma cell, the variable regions at the prong are identical to the variable regions of the B cell receptors on that same cell. So that means these antibodies that are secreted or released are going to bind to the very same epitope on the microbe that has been detected. Okay, so that means an activated B cell is not going to start making antibodies that bind to other types of epitopes. This is just giving you another view of that. Here's a little B cell right here. It gets activated, okay, because it has bound to its antigen. It's received help from the helper T cell. And in another term that you guys are going to see for an individual B cell or T cell even that has a particular type of B cell receptor, or in the case of T cells, we'll see those are called T cell receptors in their surface. Those are called clones. So we're saying you've only got one B cell around with a particular type of B cell receptor in it that binds to a particular epitope on an antigen. But you could have multiple copies of that same cell, and each of those would be considered to be a clone. So like here, when this cell gets stimulated and starts multiplying, they call that clonal proliferation. So that same cell is multiplying over and over again. You know, each of these is a clone of that original cell. And then, as I already mentioned, you know, these cells keep dividing over and over and over again, and many of those are going to specialize into those plasma cells that secrete antibodies, but then others will specialize into what are called memory cells. Those actually do not secrete antibodies, but they're very long-lived. They will last for years and years and years in many cases to help protect you if you ever encounter that organism again. Okay, so what about this additional help that we mentioned that a B cell needs to become activated and to convert into plasma cells that make antibodies? How does that happen? Okay, so here we've got a B cell clone, you know, which has a particular type of B cell receptor in its surface. Here's a whole bunch of different extracellular antigens out there floating around in some tissue fluids or in the lymph. Most likely this is happening in a lymph node. Remember, a lot of your B cells are located in lymph nodes, tonsils, other lymphoid tissue patches. This B cell has gotten lucky. It has encountered its antigen that it's very specifically supposed to bind. Okay, and then what happens after B cell receptor binds to its antigen? The antigen is brought inside the cell. That's what they're showing you here, and it gets processed. It gets clipped into pieces, and they're showing you a couple of those little pieces there as those orange triangles. And then those pieces, they wind up getting, they wind up binding to another type of protein in the cell called an MHC class II proteins. These are types of proteins. MHC stands for major histocompatibility. Can't spell today. Complex. That's a P right there, PA. Major histocompatibility complex. Histo refers to tissues. Compatibility is referring to like compatibility of tissues if you received a tissue transplant. Everybody produces these MHC class II proteins that are located in the surfaces of some of the cells of your immune system, all across the surface, and those differ from one individual to another slightly in their shape. And because of that, they play a role in tissue rejection. So when you transplant tissues that contain these particular types of cells into another individual, those MHC class II proteins are something on the surfaces of some of your cells that the person who's receiving the transplant, their immune system will react to. They see those as foreign. So that's where they got their name, major histocompatibility complex. That's how they were originally identified. They played a role in tissue rejection. But today we know that they actually have really important functions for the immune system that don't necessarily have to do with tissue rejection. And they're usually just called MHC. There's class 2 and there are class 1. We'll talk about what class 1 do later. Okay, so these little antigen fragments, little pieces of the antigen that came inside the B cell, they form a complex with MHC class 2 proteins, and then the B cell takes those MHC class 2 proteins with the little antigen pieces on them and displays those on its surface. That's called antigen display or antigen presentation. The B cell is presenting the antigen on its surface. Who is it presenting it to? It is presenting it to, let's see over here, or hopefully presenting it to this guy over here, the helper T cell, who we said had to provide assistance to the B cell to actually stimulate it to make antibodies. And again, that's called antigen presentation. We'll see more examples of that as well. Okay. And then what about these helper T cells? Okay, you've got a B cell, it's bound its antibody, brought it inside, now chopped it up into little pieces, displaying those pieces on its surface. And what it's doing then is saying, hey, helper T cell, I found my antigen, come over here and stimulate me. I need to show this antigen to you so you can stimulate me. It's trying to recruit the helper T lymphocyte over there. So what are these helper T lymphocytes? These are a special type of T lymphocyte. And now, like B cells, T cells have T cell receptors, or TCRs, in their surfaces. Now, the T cell receptors don't look, they're kind of similar to antibody molecules, but they don't have the Y shape. Okay? But they are embedded into the membrane. This is a hunk of membrane on a T cell. And at the very end of the portion that's extending outside of the surface of the cell, there's an antigen binding site, just like you have, very similar to what you have on an antibody. And that antigen binding site or groove is going to bind to a very specific antigenic determinant or epitope on an antigen. Okay? So what happens is, if there is a helper T cell that also binds to that same antigen, okay, and when that happens, the T cell gets activated, and then it is going to activate the B cell that has seen that same antigen, and it's going to do that by releasing cytokines. Remember, we talked about those a little bit before. Those are protein signals that the cells of your immune system send to each other to try to stimulate them, to try to make them better at doing their function, etc. So if the helper T cell sees that same antigen, it's going to release cytokines that will activate that B cell and cause it to specialize into plasma cells that secrete antibodies. Okay. Now, how does a helper T cell see its antigen? How does the antigen bind into that little binding site in the T cell receptor? Unlike a B cell can bind its antigens when those antigens are just floating around in a body fluid somewhere, or floating around in the lymph that's passing through a lymph node. But that's not the case with helper T cells. A helper T cell will only bind its antigen if that antigen is presented to it. Okay? Now, we just talked about that a minute ago. Over here on this slide, we mentioned that the B cell over here is presenting its antigen to, hopefully, its corresponding helper T cell that's going to stimulate it. Okay? But before that happens, that helper T cell has to have seen that same antigen separately, independently. And again, as I mentioned, that's a safeguard. You want the helper T cell to have seen the antigen independently of the B cell. Okay? And if that happens, if both a helper T cell and a B cell have seen the same antigen, that is that makes a pretty good case for, hey, this is really something that's foreign to the body and we need to respond to it. You've pulled two triggers. You know, it's kind of like the guys who, back in the old days, were sitting down in the missile silos and had the keys for the missiles that were going to go off and blow up the Soviet Union, and you had to have two of those people in there, and they both had to turn their keys in order to launch the missile. That's kind of analogous to this type of situation. It's a safeguard. Okay, so who winds up presenting these antigens to helper T cells the first time they see them? The cells that do that are called antigen-presenting cells, and those include dendritic cells and macrophages, are two very important antigen-presenting cells. Remember, we talked about dendritic cells before, the ones that have all those little, here's like the main body of the cell, and they have all these little appendages extending off of them, and you have those embedded in your skin and your mucous membranes. The reason they have all those little appendages is because those little appendages, or dendrites, are very good at capturing antigens that are flowing by. And then it brings those antigens inside the cell, and will wind up displaying those on its surface. Okay, so dendritic cells and macrophages, what they do, you have phagocytosis of something that's non-self, like a bacterium or a virus or whatever. Okay, and now this type of phagocytosis is happening as part of your innate immunity, like we've talked about before, because you haven't really made antibodies yet in a lot of cases. And as we've talked about before, you know, something gets phagocytosed, it's brought inside the cell, lysozyme fuses with it and digests whatever has been ingested. So that includes the antigens that were located there in the surface of that organism. They're getting chopped up into little pieces, and then some of those pieces wind up getting displayed on the surface of the macrophage or the dendritic cell within the MHC class II proteins. That should be MHC class II. Yeah, there we go. Inside an MHC class II protein, like we talked about before. Except you would have those MHC proteins all over, you know, located very frequently all over the surface. And those MHC class II proteins, they're going to be displaying many different types of antigenic epitopes that this particular macrophage has been exposed to. You know, it might have swallowed not only that type of bacterium that day, it might have swallowed other things as well, but it's going to be displaying pieces of the things it has engulfed all over its surface to try to activate helper T cells. Okay, and then here's a diagram showing you a helper T cell right here. And again, its surface is going to be coated in T cell receptors that bind to particular epitopes. And this is supposed to be an antigen presenting cell. This one is supposed to be a dendritic cell, but macrophages can do this as well. And if a helper T cell gets lucky and its T cell receptor binds to its own little special antigenic determinant that's being presented to it within an MHC class II protein on the surface, that's going to cause the helper T cell to get activated. Now, down here, this diagram is showing you co-stimulatory molecules. What that means is it's not quite this simple. You have to have some other signals involved that help ensure that this helper T cell is really seeing a non-self antigen and not a self antigen. But that gets a little bit too complicated. We're just going to kind of stick with what we've already talked about. Okay, so this helper T cell has seen its antigen presented to it by a dendritic cell, a macrophage, or whatever, and now it is an activated helper T cell. Oh, here's a reminder about the dendritic cells, like I was just drawing on the other slide. All those little appendages you've got there extend way out, and that just makes it more likely that those dendritic cells are going to capture invading antigens, invading pathogens that have antigens on their surfaces. Now, this is interesting, too, about those dendritic cells. Say they're located in your skin or along a mucous membrane somewhere. After they gobble up a bunch of antigens and start presenting them on their surfaces, notice this, they actually wander, they migrate to your lymph nodes and other lymphatic system tissues, like the little patches down in your intestine, and they localize themselves there because that's where your helper T cells are hanging out. So it's more likely that they'll be able to present an antigen to its corresponding helper T cell in one of those locations. Your macrophages do that too. So if they engulf antigens in some particular tissue somewhere, they migrate to lymph nodes, the spleen, your tonsils, etc., to hopefully find helper T cells to present those antigens to. Okay, so this is another diagram here. This is showing you an antigen-presenting cell, like a macrophage or a dendritic cell, has shown an antigen to its corresponding helper T cell. Now remember, we've been talking about helper T cells helping B cells mature so that they'll start producing antibodies. Now, that's one major thing that helper T cells can do. Helper T cells, though, can also provide assistance or help to other types of cells of your immune system as well, including cytotoxic T cells. But we're going to talk about what those do later. Those are really important for helping you combat virus infections. What is this help that we're talking about? That help comes in the form of cytokines, and those are protein molecules that are released by the cell. They bind to the cell that they're trying to stimulate, and they cause changes inside that cell, some sort of change in behavior, or in the case of the B cell, it's going to stimulate it to multiply and convert into plasma and memory cells. And this is actually a better view now of a stimulated helper T cell assisting a B cell that has seen its antigen. So this part we saw before. You've got a B cell that got lucky, bound to its antigen, brought the antigen inside, formed complexes between those pieces with the MHC class II proteins, and is now displaying those on its surface. And it's saying, hey, I'm waiting over here for my helper T cell so I can finalize getting activated. Okay, now here's your helper T cell. Now we just saw how this helper T cell saw that same antigen because it was presented to it by a dendritic cell or a macrophage. So now, and only the T helper cell has to have seen that antigen before to be activated itself. And only when it's activated will it wander along and find the B cell that's presenting that same antigen to it. Okay? So here you see the B cell that's seeing the antigen, the helper T cell that saw the antigen presented to it by a dendritic cell or a macrophage. They're now getting together. Okay? Because the helper T cell's receptor binds to that little piece of antigen that's being presented to it by the B cell. Now, once that happens, then, so they really physically make a connection with each other, they weren't showing you that on this slide. They weren't showing that physical connection between the B cell and the helper T cell. Once this has happened, helper T cell produces cytokines, and that stimulates the B cell to start multiplying. Okay? And many of those cells will specialize into plasma cells that produce antibodies. Evidence that a B cell has been stimulated, things that you would recognize when you have felt swollen lymph nodes before in your armpits or your, especially in your neck. The reason those lymph nodes are swelling is because B cells and T cells in those lymph nodes have been activated by some type of antigen, and they're multiplying over and over and over and over again, so that you have more of those cells to produce antibodies. Yeah, and just a reminder again about the lymph node structures in your body. These little purple, looking at a lymph node cut in half. And remember, all the incoming lymph fluid draining off of tissues that may contain microbes drains through here. And within the purple little spots they've shaded purple here, germinal centers, you got lots of B cells in there. So they're hanging out there hoping that they're going to see their antigens as that fluid passes through there, passes by those B cells. Then kind of here in the middle you have T cells. You also have macrophages, dendritic cells in there as well. The macrophages and the dendritic cells have engulfed antigens probably from elsewhere and are now in here trying to show those to the appropriate T cells, hoping they'll find the correct T cell. Then if your B cells get activated, if they see their antigens, they're going to start migrating this way. They're going to hope to encounter T cells that have been activated by the same antigen. They're moving that way to get help from the T cells. And then if they get activated and become plasma cells, they kind of hang out down in here, and they start cranking out lots and lots and lots and lots of antibodies. And then those antibodies can get into the lymph fluid that's leaving the lymph node and will ultimately get dumped back into the bloodstream. Okay? All right, so that's it for B cell activation and help from helper T cells. Next topic will be a little bit more about those memory B cells and their role in immunity.